Hello and welcome back to another of Mr. Lindsay's Biology Basics. We're going to be carrying on in the Higher Human Biology course today. We're going to be looking at the structure and replication of DNA. Okay, so this is the second in a series. Make sure guys keep checking back on the channel as I keep regularly uploading videos looking at the Higher Human Biology course. So starting off with the first part of the subunit, we need to know that DNA as a concept is made up of nucleotides throughout the entirety of the strand. Now, a nucleotide is broken into three parts. We have the phosphate, we have the deoxyribose sugar, hence the name deoxyribonucleic acid, and then we have bases. Now, it's really important as well, I need to talk about what's called a 5' end and a 3' end, so the 5 asterisk is pronounced as 5'. The reason that's the case is because deoxyribose sugar, not really shown in this diagram, is made up of several different carbon molecules. We have carbon number 1, carbon number 2, carbon number 3, carbon number 4, and then carbon number 5 sits just about here. Okay? We say the 5' end because that's where the phosphate has joined onto in that scenario, and then we have the 3' end purely because, and we're going to come back to that later, but there are bonds that will appear in this gap here. Just exactly the same from National 5 biology as well. We still have all our base pairings as well. So A joins with T, so that's adenine with thymine. And then we have cytosine and joins with guanine, so C with G. These bases are known to be as complementary in nature, and they're linked together by hydrogen bonds. Now, there's a particular reason why A always binds with T and G always binds with C. It's because of the number of hydrogen bonds that are created between the molecules. So A and T only create 2, and G and C create 3. So all these three nucleotides that I was mentioning just a little second ago, they generate what's called the sugar phosphate backbone, and that runs the entire length of a DNA molecule. So nucleotides in this chain are joined via a covalent bond. This is between the 5' end of a phosphate of one nucleotide, and it joins on to the 3' point where the carbon was I mentioned just 2 seconds ago. That sugar phosphate backbone would be a covalent bond in nature, one of the strongest bonds you can have. All right, it runs the entire length, forming what's called a polynucleotide chain. In this scenario, poly just means many, so a many nucleotide chain. DNA is also what's known as an anti-parallel structure. So when the double strand is formed, they run in opposite directions. Okay, so in the same idea, you would say a train track running along a train track are parallel, but they start at the same station and end at the same station if you were traveling in direction. Anti-parallel means they still, again, they never touch, but they run in opposite directions. So imagine one of your train tracks starting in, say, Edinburgh, the other one starting in London, and they'll cross halfway along. Okay, that's the kind of similar idea we're mentioning here. Notice as well in this scenario that our deoxyribose sugar looks like it's flipped on the strand that's running down the right-hand side just now. Okay, this is the reason. That's why it's all anti-parallel in nature. A really helpful tip. The 5' end of a DNA strand, if you're ever wondering what it is, the equivalent pentagon that's sitting there, the tip of the pentagon will always be pointing towards the 5' end. Okay, so that's just a way of helping you figure out which way DNA is going if you're not entirely sure. Finally, about DNA structure, is again a continuation from National 5 biology. DNA is a double helix. Okay, just to clarify, it has anti-parallel strands. And we need to know about this 3 and 5 stuff. So if I have a 3' end here, if I follow this strand all the way up, I will eventually get a 5' end. Same with the opposite strand. If I start at a 3' end here, and I follow this strand all the way down, I will eventually get to a 5' end. Imagine DNA as a ladder. It's a good way of thinking about it. It's a ladder that's twisted. Right, so now we're going to be moving on to the replication of DNA. Okay, it's really important that everything we've just spoke about is still going to be incorporated inside how this works now. DNA replication involves the production of 2 genetically identical strands of DNA. It's a process that's called semi-conservative replication. It's not something you need to know about. It's not stated in the Higher Human course support notes from the SQA. But what that means is when every time you make a new strand of DNA, so 2 new strands, one strand or one side of that will have part from the parent, and one of the opposite matching strand will be completely brand new made. We'll see what that looks like in 2 seconds. DNA replication occurs during mitosis. Okay, so step 1 of cell division. You have to remake the DNA. It requires a few things for it to get going. So we need a DNA template. You can't just replicate DNA out of thin air. We need primers as well. I'll come back and talk about what a primer is in a second, but basically they just help everything kind of get along. We need some free nucleotides, and this is something that's a bit of a misconception that I suppose people typically struggle with. In the nucleus, there are literally hundreds of thousands, if not hundreds of thousands, of free nucleotides just floating around inside the nucleic cytoplasm area. They're there, just ready to be used. They're not there. That's not always the case, mind you. There might be times it's higher or lower. I'm not going to go into specifics. But free nucleotides are always just floating around inside the nucleus. We have enzymes as well, such as DNA polymerase. I'm going to come back and talk about that in 2 seconds. And then we need to have a supply of ATP or chemical energy going forwards. So just as a bit of I'm going to talk about things like the leading strand and lagging strand. Now I just mentioned the enzyme DNA polymerase. Okay, DNA polymerase only ever travels along DNA from the 3' end to the 5' end. So from the 3 to the 5. It only travels that direction, which means in theory on the opposite side it can only travel from 3 to 5 again as well. There becomes a bit of an issue though when we talk about the lagging strand, and obviously that will make a lot more sense when I start digging into it. But just it's really important to understand that when I just mention the leading strand, I'm talking about the side of DNA that travels from 3' to 5', and when I'm talking about the lagging strand, I'm talking about that will be the opposite side of that DNA. So DNA replication, how does it happen? So we have it in 3 stages here. So stage 1, an enzyme called DNA helicase is used to break hydrogen bonds along between all the nucleotides on the matching strands. Okay. Now in this scenario here, in the little animation we can see just going on the side here, you'll notice it starts to split from a point. That point where it splits is known as the replication fork. So you need to imagine DNA helicase is sitting inside that replication fork, moving from the 3' end to the 5' end along a lagging strand. So it's moving in our diagram here from the left to the right. So this is, you might see them as little green icons, little lime green icons on the screen here. Primers, anneal or join. Okay, so primers are short complementary sequences of nucleotides that are pre-made that allow another enzyme called DNA polymerase to bind. A primer joins the 3' end of the DNA strand. So their top strand here is the leading strand. As we can see, only ever one lime green section appears, but along the bottom, multiple lime green sections appear as we're going along the DNA chain. Okay, so our leading strand is all made at once. Our lagging strand is copied in little chunks. Now, DNA polymerase is really important, guys, that we make sure if we're ever answering a question from the SQA that we say DNA polymerase, because there is an equivalent RNA polymerase. So we can't just get away with saying that. So it would be good practice to just say DNA polymerase. This DNA polymerase adds free complementary nucleotides. So if I had a chain that went AAA, TTT, CCC, GGG, it would match in TTT, AAA, GGG, CCC. Okay, so it just matches them up as it sees them. Okay, has a remarkably high success rate. Okay, we're talking very, very infrequent mistakes. The leading strand, again, I've said previously, is synthesized continuously, but the lagging strand is done in sections. And the reason it's done in sections is because this process is continuous. DNA helicase is traveling along DNA at the same time, almost like you're unzipping the zipper on your jacket. As we've always said as well, DNA polymerase can only travel 3' to 5', but as we can see on the lagging strand, the 3' to 5' direction only opens up slowly over time, where the leading strand, the top strand, opens up continuously. So DNA polymerase joins onto those primers in the lagging strand, all the different primers that are there, and he makes these sections that are called Okazaki fragments. These Okazaki fragments are then joined together by a DNA ligase, like a type of glue. Okay, so that's a little like poof animation, the little pop you see on the screen there. The last part of this subunit we need to look at what's called a polymerase chain reaction, which is also known as PCR. Okay, so PCR is used to create many copies of a piece of DNA in a lab really quickly. To amplify a specific sequence, primers are used very similarly as we just saw in the last scenario. It allows things to join on. But these primers are more specific, so they're complementary to a very specific target sequence, so you can only get the bit of DNA amplified that you want, and you get lots of copies of it. So not just the entirety of DNA, only the sections you want particularly. So ultimately, what is PCR? Okay, so PCR is a process we've said that carries out DNA replication in a lab setting much faster than it happens in the body. There's a variety of different steps we need to know about this when we're going through. So to replicate DNA in a lab, you would need basically to go through these following steps. So first, we need to heat the DNA to 92 and 98 degrees. This does the same job as DNA helicase, but instead of it being a continuous unzipping process, every single hydrogen bond just breaks down. So the entire two strands just separate immediately. We then need to cool the DNA to roughly between 50 and 65 degrees, and this allows specific primers to bind. Remember, those specific primers are complementary. So again, A binds with T, G binds with C, the exact same concept we just covered 2 seconds ago. And in the third part, our temperature of the whole thing is raised to between 70 and 80 degrees for heat-tolerant DNA polymerase. It might be seen as, it might be reference places on the internet known as TAC polymerase. And this does the exact same thing as the DNA polymerase we just mentioned. It adds free nucleotides to the new strand using the DNA molecule we've got as a template. Then this process just repeats and repeats and repeats. So in the previous little section I was just talking about, you saw an image of what was known as a thermocycler. The thermocycler is just a piece of kit that basically runs this process, and then basically each cycle would take 3 minutes roughly. So every 3 minutes, you've created another copy. And remember, it divides into 2 every time, so 1 becomes 2, 2 becomes 4, so on and so forth. So last part about this then, we need to know about the practical applications of PCR. Okay, so PCR can be used in many different ways to help understand many aspects of human life or come to understand things that have happened. So we can amplify DNA to help solve crimes. Okay, so a blood sample's left behind or whatever, there will be DNA left there. You can amplify that DNA, then match it to an individual, whether that be a victim or a criminal. You can be used to settle paternity suits. So similar concept, you can take the DNA from a child that has a disputed parent, and then in theory, if the father is asking to find out if it's his child, half of the child's DNA should be his. So we can amplify the two together, then try and match them up and see if that works out. We can then use PCR to help diagnose genetic disorders. So try and get ahead of the curve, make sure that we're seeing, whether it be in animals or in humans in particular, we can try to understand if there are any genetic disorders on the go. Right, guys, well, that brings us now to the end of subunit 1.2. We'll look at the structure and replication of DNA. It meant a lot to me. If you liked this video, leaving a like at the bottom gives me a good indication, a good bit of feedback if this is a video that's landed well with you guys. I'll be moving on again with the Higher Human course. Next video, we're going to be looking at gene expression. So hopefully I'll see you there, and I'll catch you next time.